Welcome to the SB Grid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SB Grid.
Hello, everybody. Thank you for joining. This is Jason Key at SB Grid. Happy Pi Day. Today is March 14th, Pi Day, so celebration of mathematics, and I take it as a celebration of science in general. Pi Day is a big holiday in our family. We always make pie. So with that, it is my pleasure to introduce Kirash Jamali, joining us from the MRC Ensure Shares Group. Kirash, thematically appropriate for Pi Day, has a background in mathematics and sort of has taken on computational approaches to structural biology problems. And so I guess the first application born of those efforts that I've seen is ModelAngelo, which he's going to tell us about today. So Kirash, go right ahead.
Thanks. So yes, I'm Kirash Jamali in the Shares Group at the MRC Laboratory of Molecular Biology in Cambridge, United Kingdom. And today I'm going to be talking about ModelAngelo, which is an automated atomic model building program for cryo-EM maps. So I'll get started. There we go. So this is a cryo-EM map, and we want to build this atomic model into it. And the way this is normally done is, if you want to do it de novo, you basically kind of just do it residue by residue manually. And so we wanted to automate this process in an end-to-end fashion. And so what we came up with is ModelAngelo, which is a three-step process to build atomic models. And the three steps are basically, you first start with C alpha atom predictions, which is kind of like to place each residue separately just from the cryo-EM map. And then you take the sequence information as well, and you try to build the full atomic model from those initial points, as well as the cryo-EM map. And then afterwards, we have post-processing to fix some issues. So for the C alpha part, which I won't really be talking too much about today, is, you have your C alpha atoms, they're in this cryo-EM volume, and we voxelize it. So we basically ask the question within a 1.5 angstrom kind of cube of the cryo-EM map, is there or is there not a C alpha atom existing there? And then this can become a machine learning segmentation problem. So basically, you have your cryo-EM map that goes as an input, and the output is just a bunch of probabilities for each voxel, whether or not it includes a C alpha atom. And so, here's your map, and this is real data. So that's an actual map that we give ModelAngelo, and on the right for each orange dot is a C alpha prediction it has made. But let's assume that this part we have already trained the network, and we want to move on to how to build full atomic model from this. And the way that works is basically the heft of what ModelAngelo is, which is this graph neural network. And we had to specifically design it so that you're able to combine all these different pieces of information together, which is the cryo-EM map, which is a 3D, voxel grid. You have the sequence, which is just text, the amino acid sequence for all the sequences that you expect to see in the map. And then you also have your initial C alpha positions, which is just a graph of positions. And how do you combine all this data together is actually difficult and has never been done before. So we designed this graph neural network, which basically takes each module is responsible for extracting information from a specific piece of these inputs, and then it's all integrated together within the graph. And each module is based on this attention algorithm that has now been taking all of machine learning by storm. So if you've ever used ChatGPT, AlphaFold, all of these all use this attention algorithm. So here it is. And what the attention algorithm is it's a differentiable search procedure. So you have some object, you represent it with a vector using by putting it through a machine learning, let's say MLP, and then you get a vector at the end. And then you also have a library of things that you want to compare it against. And so what you do here is you do basically a dot product of this vector to your library of vectors. You get a similarity, and then you marginalize over these similarities with a different representation of the library. And what this gives you at the end is, for a new object that has come in, what kind of information can I extract from the library of things I've seen in order to get a new representation for the object based on how it relates to the library. That's a bit abstract, but I will crystallize it a bit when I go through each of these modules. So for example, the CRY-M module, what it does is it looks at each of those C alpha initial points that I talked about. It takes a cube of density around this, right? And you've probably heard of convolutional neural networks. This is how, let's say image processing with new machine learning methods works, and also 3D volume stuff as well. And here in ModelAngel, we use it as well for the CRY-M module. So you interpolate the cube around each C alpha, and the cubes are also kind of oriented based on the backbone orientation at the C alpha position, which you might be wondering, how do we even get the backbone orientation? And in the very beginning, it's completely random, but then ModelAngel will update this backbone representation as it goes along. So we interpolate this cube of density around the C alpha atom, and we put it through a convolutional neural network. We get a vector. We do similar for rectangles of density between the current C alpha atom and all its nearest neighbors. So the 20 nearest neighbors, we kind of look at rectangles between them. And this is supposed to give it an idea of whether or not there's a peptide bond to C alpha atoms. So then this also goes through a CNN, and we also get another vector. And then the cube kind of represents the query, so the object that comes in, and all the rectangles represent the library of things. So the idea is to say, for this C alpha atom, which of the other atoms around it does it actually connect to? And then based on that, we can actually get the information from those nearby atoms to come into the current atom and kind of update the representation. The sequence module, which is the second module that goes after, then asks the question, okay, for each residue that we have a C alpha atom for, given the representation that, let's say before, it's been updated from the CRY-M module, can we see anything between this and the sequence that came in? And so the sequence that's just text goes through a protein language model. So we use the ESM language model from Facebook, and that turns it into a series of vectors. And then we do a similar search against the sequence, and we calculate similarly to the sequence, and then we bring it back. So this is basically now corresponds. Before we just had CRY-M information, now you also add in the sequence information from the amino acids that were inputted, and you get an updated representation. And lastly, we also do this module, the spatial invariant point attention module, that is very similar to the one that's in AlphaFold. And the idea here is now you want to integrate information about the geometry of nearby residues, not just the CRY-M information and not just the sequence information, but also the geometry, whether or not something's in alpha helix, beta sheet, etc. And the way this works is, for each residue, you kind of predict points around that you should look at. And then if there's a nearby residue that is close to that point, then you get a high weight, high similarity from that residue when you bring in information again. Okay, so in this graph neural network, once the information goes through each of these different modules, the representations keep getting updated for each residue. And then using these updated representations, we put it through this backbone frame module. And what this one does, for example, this is one of the predictions we make, is that for this backbone frame that we talked about, which is just a triangle with the C alpha atom, the nitrogen and carbon atoms, it predicts how much to shift each of these atoms, right? So, in the beginning, when we did the segmentation task, if you remember, it was like a 1.5 angstrom voxel. We just said that the C alpha atom exists in this voxel, but that doesn't really tell you anything about where in the voxel it could be. So the C alpha atom itself starts off a bit wrong, and on average, it's like 1 angstrom off. And then the nitrogen and carbon atoms we just started them off in a random rotation. So those are completely wrong in the beginning. But then every time these features are updated and they go through this backbone frame module, we get these shift vectors, and then now we can actually update the orientation of this triangle. So each layer of the graph neural network updates the orientations, and we get closer and closer to what the actual true orientation of this backbone is. So this is one of these modules, but there's a few more. There's a torsion angle prediction for the side chains. There's amino acid prediction probabilities. And so all of those are predicted after each of these modules has updated representation. Okay, one of those things is also a confidence measure. This is very reminiscent of the AlphaFold confidence measure, but it's based on the backbone RMSD that we expect to see. So basically, if the cryo-EM volume at some point has a low local resolution, Model Angelo kind of knows this, and it knows that it can't confidently place the backbone. So it gives you a lower confidence, predicted confidence for how this loop is going to look, for example. And that is represented in the B factors that in the Model Angelo output file, which we think is very useful. So that's also predicted by the graph neural network. So for the post-processing, once everything goes through this graph neural network, we need to actually take this and turn it into an actual atomic model. So what we have is we have probabilities for an amino acid, which amino acid type for each residue. We have, and it's not entirely clear how you make the statement of like which amino acid it actually is, because we can take the highest scoring amino acid that Model Angelo thinks it is, but then you're not really using the information that you got from the sequences that the user has provided. So the user knows what sequences exist in their map most of the time. And if you just take Model Angelo's highest predictions, you might actually end up with wrong sequences. And so the way to fix that is to actually, instead of taking the highest scoring sequences, which is what people normally do, you want to take the probabilities that Model Angelo is actually predicting and use those to somehow pick where in the sequence it is. And the way we do that is we basically turn this into a hidden Markov model. And so basically each amino acid that we have the probabilities, and then we create a hidden Markov model profile, and then we do a search against the sequences that the user has provided. And so there you go, that's the hidden Markov model. The transition probabilities, as of right now, are just average transition probabilities, but just taken from literature. But actually, with the new Model Angelo version, which I'll talk about later, we've made this better. So you take this hidden Markov model, you search it against the sequence file with the HLM line, and then you get much closer amino acid predictions than you had before. So more correct ones. And so this is an example of a file we built, a model we built with the sequence. So the black outline is what's deposited, and the blue is what Model Angelo predicted. And you can also see that using the torsion angles that Michelangelo predicts for the sidechains, we actually more or less get good sidechain orientations predicted. And now the thing is, this is before refinement. So you actually will, if you use ServoCAD or something, these sidechains orientations become much better. So we also wanted to see whether or not we can do this without the user providing sequences. And we know that if the resolution is high enough, when people are building things de novo, a lot of the time they can make some, judgments about what they think it is. So we wanted to see if that's possible. And so if you actually train the graph neural network itself without having the sequence module, you are then able to run Michelangelo without sequences. And then we wanted to see how that performs. So this is for an amyloid in the Sherry's group. We also work a lot with amyloids. And this is TMEM106B, which had like a very high resolution map, but they didn't know what protein it actually was. And they spent months on this and to find out what it was just by basically looking at the map itself. And this was much before Michelangelo. So we wanted to see if you could potentially use Michelangelo for a purpose like this. And so this, the model you see here is built by Michelangelo. And how I mentioned before that if you just take the highest probability without the sequence, if you take the highest probability amino acids, and you can see that already it's pretty accurate because the resolution is very high here. And then if we do the same thing and we build a hidden Markov model based off of this, and then instead of searching it against a human provided sequence file, we search it against all of the human genome, you can actually see that you also find TMEM106B with a high confidence. So you are able to also use this to find sequences that you might not know exist in your CRY-M map. And so the conclusion for this is that we now have a novel way to encode CRY-M density information in graphs using for machine learning purposes. It results in automated model building of proteins when the resolution is high enough, so up to 4 angstrom. Of course, as you get closer and closer to 4 angstrom, the results get worse. And also, even if your map has a high global resolution, if the local resolution is bad, it's not going to do as well there. And it's also capable of resolving unknown sequences when the resolution is high. So when resolution should actually be a bit higher if you wanted to get things that are unknown sequences. So how do you actually use this? And this is what I gather the seminar is mostly based around. And the command line interface for Michelangelo, we strive to keep it very simple. So there's actually 2 main ways of running Michelangelo, which I said before. So it's with the sequence and without the sequence. So if you want to build it with the sequence, you just run Michelangelo build. You give it a volume, a path to the volume, and you give it the path to the FASTA file. And actually, a lot of the time when people have bugs, it's because of the FASTA file. So please make sure it's formatted correctly. And then you also give an output directory. And what the output directory structure, once it's done, looks like is something like this. So you get your output directory, and then inside are 4 folders. So the C alpha output, and then a GNN output round 1, 2, and 3. So I didn't really mention, but the GNN, it has 8 layers. And then once it's gone through the 8 layers, that's basically 1 round of the GNN. But then at the very end of it, you have an atomic model. This is closer to the real atomic model than when it started. So then you can apply it again, and then you get closer and you can apply it again. And then output round 3 is basically the end stage. But these are intermediate results. You probably, most of the time, you don't really need to look inside these folders unless you want to debug something. So the 2 CIF files that exist in your output directory are actually the outputs. So we have the output.cif and we have output raw. Output .cif, when you build with sequences, is pretty aggressively pruned based on the sequences provided. So if Model builds something and is unsure of the sequence, or if it's sure of the sequence, but the sequence does not exist in the FASTA file, it will prune it away. Those still exist in output_raw. So if it hasn't been pruned away in the raw file, that could mean two things. One, Model was not very certain about the actual sequence. So it predicted one kind of sequence, or it had a low prediction confidence for a portion of the sequence, and it just wasn't in the sequence file. And so that could either happen if the local resolution is bad, or if you've forgotten to put the sequence in there, or that specific sequence in there. Now, if you want to build without sequence, it's the exact same. It's just the command changes to build no seq, and then you don't have to give it a sequence file anymore, thankfully. And then what the directory structure looks like then is the same intermediate folders as before, but now you have this other folder called HMM profiles. HMM profiles is a folder that for each chain in your output.cif also has an HMM profile file in it. And this is actually what we use, for example, for the TMEM case, where you want to search against a genome or something, you would use these profiles. And I'll explain how. But then the output.cif here is actually the same as the output_raw in the build with sequence. So it hasn't been pruned because you can't prune anything. You don't have access to sequences anymore. So it's a bit more messy than the normal Model. How would you actually do a sequence search? Currently, the best way to do it is to use HHBlitz. So the HMM files, HMM profile files that we provide currently in Model 0.2 are HHM files, which are HHBlitz profiles. And you have to install HHBlitz, and then you would have to also use their kind of database files. It's a bit difficult to use. And then you run a command like this to get access to your results. But it does work very well. And so some common issues. One really big one, especially if you think your map is very good and the result just looks horrible, it's that the map is probably in the wrong hand. So first thing to try is just flip the hand of the map, try it again. If the result gets better, that was probably why. Another way to diagnose this is if you look, like in Chimera, for example, if you look at the confidence values Model predicts, there should be some high confidence chains predicted. So if there's no high confidence chains predicted, yeah, it's probably in the wrong hand. So that is the main, most common issue we have. Second one is if you see a lot of pruned away sequences in the output.cif, but in output raw, it still exists. Either it's a problem with the local resolution, but if you definitely think it's not a problem with the local resolution, then it's highly likely that that sequence is not provided in the sequence file that you provide. So to diagnose this, you could, for example, run Model without the sequence, then take the HMM profiles, do a search against like whatever genome you think, and then look through the sequences. If you see that, yeah, there's a new sequence in here, that could actually be the problem. The third problem is that the global resolution is too low. So, you know, if it's not going to automatically just stop working at 4 angstrom and above, but it will get worse, and it'll get worse pretty quickly. And, you know, it's going to perform best at 3.5 and higher resolutions. Similar problem is the local resolution. So even if your global resolution is high, the local resolution, if it's low, you know, it's just not going to be very confident, and it's not going to be able to distinguish the sequence and so on. And the last one is, yeah, sometimes it can build a confident backbone This is when like the local resolution is on the cusp of something where it can see the backbone, but it can't build the sequences, even if you've provided the sequences. And yeah, this happens sometimes, and it's really hard to do anything about it because just the sidechain densities are probably not good enough for Michelangelo to understand what's happening. So to diagnose this, it's just you're confident that the sequence exists in the input, and Michelangelo raw, like the raw output, has this that chain, but it just doesn't exist in the pruned sequence. Then you could still use the backbone that Michelangelo has predicted, but you would have to go and fix the sequence yourself. So yeah, those are some of the common issues. So what are next steps for Michelangelo? Well, we're going to have the 1.0, which we will release soon. And there's a lot of improvements. The first one is that everything I've explained so far is for proteins only. But we are going to now also have the ability to do nucleotides. Nucleotides are considerably more difficult to build automatically. And so the results won't be as good, especially for lower resolutions as the proteins, but it should still be respectable to build the backbone. And when the resolution goes very high, like 2.5 angstrom, then you could really actually do a pretty good job of completely automatically building it. Michelangelo currently 0.2 downsamples everything to Nyquist of 3 angstroms, and because we had some issues with the nucleotides, 2 angstroms works much better for the nucleotides. So that's what we're going to be using for Michelangelo 1.0. This actually also slightly improves performance on proteins as well. And we will have an integrated hammer search, which is going to be very useful for Michelangelo 1.0. So instead of having to install HHBlitz and dealing with all that, we're going to have an integrated utility that also understands how Michelangelo HMM profile directories work. So you can just finish your build no sequence job and then just do an HMM search job right from there, and it's going to understand everything and parse everything correctly, and it's going to make your job of just searching genes like a genome much easier. So that's going to be in Michelangelo 1.0 as well, and it will be quite a bit faster, the whole thing, because of some optimizations that we've done. And yeah, so please try it out if you haven't already. There's instructions available in the GitHub repo. And also, if you just type Michelangelo build and then help, it will give you all the options that are available for each command. And yeah, so I'd like to acknowledge my supervisor, Shor Seres, Dari, who's the co-author on this paper, other members of my team, and also Lucas Shell, who helped with the HMM. And yeah, I can take some questions now.
Great. Thank you very much, Jirash. For questions, you can raise your hand. I'll give the little clap symbol here. But if you go to reactions and then click raise your hand, big button at the bottom, hands will pop up and it will bump you up to the top of the list. And then I can unmute you and you can ask your question directly. You can also send questions in the chat window and we can just pass those on. Just a thought as you were wrapping up there, I can tell you that when I installed Michelangelo, I installed it, it's available in SPGrid. I think we have 0.2.2 is there. I picked a sort of arbitrarily hard map and just ran it as my test case. The very first time it ran through beautifully, built a very nice little model, and and it ran great. And I can't say that for many other software packages. So that's
Thank you.
that's great. One question that came in by chat. We observed that the chain was built, but in fragments. We tried after changing the map orientation, but still observed the same result. Can you suggest something regarding the same? The map local resolution between 3 to 5, much of it in the 3.2 range.
Right. So yeah, if it's fragmented and you could take a look at the raw file. So if that one isn't fragmented, that's probably what's happening is in the post-processing script, it's done a search. So it can see some portions of the sequence that are confident in, and other portions where it's amino acid probabilities are getting less confident. So it's going to prune those. So there's not much to do other than just take the chain, and if you especially if you think that the sequence exists, not much to do than just kind of fix it manually. It's probably just the local resolution at those specific points is not good enough for Model Angelo, or yeah, something like that.
All right, we've got hand up Chao Kwan. You can unmute and ask your question.
Thank you. It's great talk. So yeah, I'm actually working on a case where it's difficult to dock a model into the map in Chimera. So when I look at the map, I find the map quality is pretty poor. I mean, resolution is good by good face value, but the quality is poor, probably have a preferred orientation. Yeah.
Okay, right.
So yeah, I'm wondering, in this case, do you think that your software can help, like at least can you?
So what is the resolution? Just global resolution number?
It's 3.2. Again, it's just the face value, so the quality is actually very.
You can try it. So what it's probably going to do is, like, for a portion of it, it might be able to build it.
Okay.
But yeah, if you already think it's not very good, it's So basically, Model Angelo, when a human can build it, we can build it faster. That's the idea, and easier for you. But then if you are having like trouble, it might be that Model Angelo can't do it either. But you should try it and let me know. If it does build it, that's great.
Certainly hope so. I mean, I'm thinking, like, because the first step here is localization of the C alphas, right? So
yes,
if you have localized the C alphas, then it should help. In theory, it should help localize the secondary structure features. So that's going to be help. Yeah, that's my guess. Yeah.
So if it's able to do that, and you can actually, so we put the intermediate files there for a reason. So if you know, this is a more like advanced whatever, but if once it runs, you can look in the C alpha output folder, and then there's a CIF file with the C alphas in there, and just look. You know, that might even help you, even if Model Angelo itself isn't able to do anything afterwards. It might help you see where things are. So
cool. Thank you. Appreciate that.
We had a question here from. I noticed that building into maps in which symmetry has been imposed, built chains that were not identical. Let's see. Imposed symmetry. So can symmetry be accounted for without user map segmentation?
So basically, Model Angelo will basically build everything independently, almost assuming there's no symmetry whatsoever, and you will actually see specifically regions. And this comes up more in when you want to do searches against the genome. But like, you know, it's the exact same symmetry, I understand, but for some reason, Model Angelo, and the reason is like, it's a bit technical, but it will have like better chains in one symmetry section and not the other. And I don't know, just you can duplicate it or something, but
Choose the best chain and apply symmetry.
Exactly. Yeah. So.
One other question: is there a size limit for Model Angelo for building a model?
No. Yeah, we've built things down that's like 160,000 residues. It takes longer, but it should be fine. Yeah.
And I think you touched on this. Your resolution, you're aiming for about 3.5. You said is sort of ideal for the best quality model.
And better. Yeah.
Say that again. Sorry.
And better. So,
and better. Okay, so it drops off pretty quickly after 3.5.
Yeah. It's like mainly the side chains that go first, right? So, you know, it's
So we had a question: which resolution of the map can get a good model? So 3.5, and then you're living dangerously
Yeah, exactly.
I think.
Yeah.
Pete had a question. Go ahead, Pete.
Great talk, and I've actually got two questions, and they're both a little bit speculative kind of ones.
Mhm.
But for the first one, so do you have a sense for how sensitive it is for the density type? Like if you wanted to use this for a low resolution X-ray map, or if you had a low resolution micro ED map, would you have to
Yeah.
retrain that portion of the model, or you think things would be close enough?
So I've been asked this before, and I know that there are other machine learning approaches that work with densities where apparently it doesn't need to be retrained, but I personally have never tested Model Angel or anything else. So I really couldn't say. My intuition says it should be retrained, but I don't know. Yeah.
Not knowing is a perfectly valid answer, and it's speculative. So it's, and I guess the other one might be a little bit more speculative. But for the unknown sequence case, have you thought about using, like, if you've got a three-dimensional structure that you don't know the sequence of, would doing a search with that three-dimensional structure rather than sequence
Right.
probability, is that something that could be useful?
Yeah, that could be useful. So like FoldSeq, for example, does something like that. But basically, we've seen much better specificity and when we use the sequences. Because if you think, like, Model Angel has, like, it's predicting two things. Like, it's predicting the structure, but it's also predicting these probabilities. And
Yep.
there's a lot more information in the probabilities than, because structure is conserved much more than sequences anyway, right? So if you want to find, yeah. So
Cool. Well, thank you.
Any other questions? Anyone, feel free to raise your hand or send them by chat. All right, great. I mean, I have a question. So, you're coming from a mathematics background, you're tackling different problems in structural biology. Like, this is a challenging one, and this is a pretty productive approach, at least in my hands. What are your targets on next? Are you improving this? You're going to nucleotides. So
yes,
that's interesting.
So yeah, we're going to be improving this for now, and I'm kind of somewhat involved with other things for cryo processing itself. But after that, who knows? There's some other projects, but they're pretty preliminary now. So
Yeah,
I'll let you know.
all right, cool. Yeah, that's great. I just I see there's so much rapid development happening in this space. So it seems like there's so many great ideas out there. Just good time to be developing methods in structural biology.
And use them, hopefully.
Yeah, great. All right, well, with that, we can wrap up.